in, but thank you everyone for joining us today. Depending on what time it is where you all, good morning from my end. For our next installment in the Amplifying Voices seminar series. We have some really exciting speakers lined up for today, all of whom will be presenting their work focused on aquatic plants, hence the special session today. So I'll keep my introductory remarks short. We have a lot of exciting work today. So I would just note, if anyone has any questions for the speakers, I'd invite you to either type them in the chat during their presentation. We'll be keeping folks muted, or wait until after they finish presenting. We will have time for question and answer sessions following each of the presentations. So we'll make time for that after each presenter as well. So with that, I'd like to introduce our first speaker, Vanja Milovanović from the University of Belgrade in Serbia. Ms. Milovanović is an aquatic biologist who received both her Bachelor of Science and her Master of Science at the University of Belgrade, where she was recognized with an award for Best Master's Thesis. She's currently pursuing her doctorate in biology, also at the University of Belgrade, and today she will be presenting some of her graduate work examining charophyte presence in river floodplains and their potential for informing restoration efforts. Welcome, Vanja.
Thank you. Should I share my screen? Yes, of course. Do you see my presentation?
Yes. Looks good on our end, thanks.
I will try full screen.
Yep, that's working.
It works
just fine now.
Great. Okay, now, hello everyone. My name is Vanja Milovanović, and I am a PhD student at the Faculty of Biology, University of Belgrade in Serbia. So for those of you that maybe don't know where Serbia is actually a country, a beautiful country in Europe on the Balkan Peninsula. Its capital city is called Belgrade, and it's the place where I was born, where I lived my whole life, and where I am now doing my PhD. So here you can actually see the main entrance to the botanical garden Jevremovac, where I am actually now. Here you can see the administrative building, and if you go down the stairs, you can actually see my lab or my working place where I am spending most of my working hours. So first of all, I would like to thank to the Association for the Sciences of Limnology and Oceanography for inviting me to be a part of this amazing Amplifying Voices webinar. And I would also use this opportunity to thank to the Rafford Foundation for supporting our work, and especially my PhD project. Today I will talk about Charophyte oospores and gyrogonites in recent sediments of small water bodies as taxonomic characters and indicators of revitalization potential. First of all, what are actually charophytes or stoneworts? Where they are macroscopic large algae that build dense underwater meadows in stagnant or slow-flowing brackish or fresh waters. And there are currently registered 27 species in Serbia that belong to 5 genera. So Hara, Sperohara, Nitella, Tolipella, and Nitelopsis. And they are important as they provide the base of a food chain in aquatic ecosystems. So they actually provide the food for water birds, also a safe place for fish, also for zooplankton and for some other invertebrate populations. And also they act as nutrient sink and enhance water clarity. Traditionally, morphological characteristics of their thalli, such as cortex type or arrangement and appearance of spines on the cortex, as well as morphology of the stipular wall, have been used to identify charophytes to the level of species. However, this is not always possible. For example, there are some, or this could be actually very difficult. So, for example, there are some taxa that are morphologically very similar in their thallic characteristics, and also sometimes the variability of these characters can be very pronounced. For example, due to the effect of different ecological factors. So in all of these situations, actually an alternative may be oospores and gyrogonites that are actually trapped inside the sediments. So what are actually oospores and gyrogonites? Those are reproductive structures of charophytes. So oospores are charophyte zygotes, and gyrogonites are actually nothing more than calcified oospores. And they can be trapped in sediments, and then they are the constituents of so-called diaspore bank and are called diaspores, and they are available for sampling and analysis even when the thallus itself is no longer available. Identification key for charophytes based on their oospores already exist and are regional. So, for example, for Central Europe or New Zealand. But if we talk about Balkan Peninsula and about Serbia, then no such key exists yet. And if we talk about gyrogonites, those of fossil taxa are pretty well described. But if we talk about recent or extant taxa, then this is actually not the case. On this slide, you can see basic morphometrical parameters of oospores that I have to measure. Those are oospore height, oospore width. I count number of ridges, also measure the distance between two ridges called fossa, and also there are some qualitative characteristics that are important, such as the appearance of the basal pole of the oospore and also the presence of basal cage, claws, and plug. And on this slide, you could actually see the comparison between oospore and gyrogonite of the same species. So you can see that they clearly differ, and there are even some indications that the parameters of gyrogonites are even more taxonomically significant than those of the oospores. Also, qualitative characteristics of gyrogonites are important, such as the configuration of the apical pole, configuration of the basal pole, and also basal plug. So to get back to my research has some main aims. Those are, first of all, to assess the degree of variability of diaspora morphometric parameters between different populations of the same species, as well as between different species, in order to establish the taxonomic significance of these parameters and the potential for precise identification of charophyte taxa. Then to assess the potential influence of physical, chemical, and hydromorphological parameters on the variability of diaspore parameters, as well as on the phytosociology of charophytes. To, of course, create the first local key for the identification of charophytes based on standard taxonomic parameters, as well as oospores and gyrogonites in Serbia, which will also contain data on the phytosociology of the species. and to assess the viability of oospores and gyrogonites using a crash test, and to give preliminary recommendations for the preservation and revitalization of their habitats in the researched area based on actually composition and viability of the diaspora bank, all in order to, of course, preserve and restore native diversity. Description of the localities. This project actually takes place, of course, in Serbia, in the floodplains of three rivers. Those are Danube River, actually on the territory of the Ramsar site Labudovo Okno. Then there are also Karaš and Nera rivers, actually on the territory of the area of outstanding features Karaš Nera. So this is also the protected area, and some localities are actually in the non-protected area that is in between and nearby those protected areas. And two out of these three rivers, Danube and Nera, are actually designated as important international ecological corridors, while the third one, Karaš, has been intercepted by building Danube-Tisa-Danube channel, which actually led to forming numerous oxbow lakes. But anyway, according to their type, my localities are actually, gravel pits, sand pits, and fluvial lakes or ponds. Now, field work can be divided into four groups. First of all, georeferencing of localities, so measuring of physical and chemical parameters of water. Then phytocenology, which actually means collecting the data on the composition of Charophyte communities. Then a lake habitat survey, which actually means collecting the data on the morphology, pedology, anthropogenic pressures, hydrological regime, and sediments at the level of so-called LHS hub plots that are positioned at the same place where phytocenological data are collected and where sediments are also collected. And sediments are collected using tube-like sediment core with the diameter of 4 centimeters. And we chose to do the methodology of forming replicates that could consist of 3 to 5 pseudo replicates in order to, yes, because actually literature review showed that it is better to collect several smaller samples than one large, all in order to actually improve the accuracy of the assessment of the number of diaspores. And yes, each pseudo replicate actually consists of up to 3 sediment depths, so up to 5, up to 10, and up to 15 centimeters if possible. And laboratory work actually involves identification of individuals and also storage and collection of mature oospores in order to be used as a reference base. Then process of sediment drying in adequate conditions, and also sieving through different mesh size in order to form fractions that are actually being observed under the stereo microscope. And then is the step of diaspora extraction and detailed photographing and measuring. and the classification of diaspores into categories, after which I take several of them in order to make scanning electron micrographs, and then viability test, crash viability test is performed. This test distinguishes two classes of diaspores, so apparently viable and apparently unviable. Apparently viable are those that actually resist to crushing while being pressed with forceps, and of course unviable are those that actually show no resistance to crushing. Then is the step of data processing, so selection and application of statistical analysis. And these are some expected results. I will just quickly go through them because I hope to actually met all of my aims. So actually the potential of independent application of morphometrical parameters of diaspores in the identification of Charophytes to the species level will be assessed. Then the influence of habitat characteristics on the variability of oospore and gyrogonite parameters among different populations of the same species will be assessed. The first local key for the identification of Charophytes based on standard taxonomic parameters, as well as oospores and gyrogonites in Serbia, will be created, which will also contain data on the phytosociology of the species. And last but not the least, the viability of oospores and gyrogonites in the diaspora bank at the investigated localities will be assessed, and preliminary recommendations in the researched area will be formulated. Okay. Thank you all very much for your attention, and I would also like to thank to the Ministry of Science, Technological Development and Innovations for supporting me with scholarship, and that's about it from me. Thank you again.
Thank you, Vanja. That was wonderful. If anyone has any questions, feel free to raise your hand. We can try to find your video, or if you would prefer or having connection issues, feel free to type the questions in the chat.
I would like to ask the question. I'm glad that also charophytologists have a representation in this series of webinars. It is wonderful because I'm also representative of this small group of researchers. And my question is related to your field study. How deep were studied sites?
How deep? Well, those are all like shallow water bodies. Sorry, I have a stuttering problem, so sometimes I hope that you can understand me. Anyway,
yes, sure.
Okay. Yes, those are all shallow water bodies. So some of them were like really shallow for about like half meter to 1 meter, but there were also some that we actually call lakes here, that were actually up to 5, but sediments were collected only in the shallow part. So I don't have the instruments to collect at the deeper part of the lake.
Okay, and I have a next question. It's also related to the sampling process. And did you collect the sediment scores from the Charophyte meadows, or from the sediments free of the vegetation, or from both type of sites?
From both, because sometimes I even didn't know if the Charophytes are there. You know, I just wanted to check if they are present. Yes, and because, sometimes yes.
Because if Charophytes are in the place which you decide to study, it is obvious that probably oospores are there, but in the
Yes.
sites without vegetation, we are not sure about this. So sure. And
Yes.
are you planning to do in the future some kind of experiment or mesocosm experiment?
Well, currently no, but in the future probably yes, because right now I actually have to finish my PhD project.
Yes.
I have some several years to finish it, and after that I will be free to do anything
Yes, sure.
else.
Because
Yes.
maybe it would be also good to testify the Charophyte viability in the laboratory, to have broader knowledge about the possibility to possibility to regenerate the vegetation from the oospores, because it is one
Yes.
of the most important for us as a hydrobotanist, to know if it's high or low potential for vegetation regeneration in the future, because it is really important. So thank you for a wonderful
Yes,
talk.
it is. Thank you very much.
Thank you. I actually had some questions similar to that. This is really interesting to learn about. I don't know much about charophytes myself, but when thinking about the diaspora bank, I guess I was curious to know how long, I guess, what is the length of an average charophyte's life cycle? And then how long are the oospores in the diaspora bank typically viable for?
Mhm.
Is
Oh,
there, do you
yes.
have any sense of that?
It's a pretty good question, actually, where literature sources are different about it. Mostly they say that they can, I think about up to several decades, you know, but somewhere I even read about up to 300 years or something like that, but I am really not sure about it, so
Okay.
please.
So I have the answer to this question.
Okay.
In the case of the previous study, the group of Professor Maria Rodriguez from University of Valencia studied this topic, and even charophyte oospores from the sediments older than 100 years were able to germinate. And it was published, if I'm right, in Aquatic Botany. But the most important question is the fact that charophytes are able to germinate only in the quite shallow sediments. I mean about the sediment layer.
Yes.
This layer is not wider than 7 centimeters. It is probably 5 centimeters or 6 centimeters, because if we would like to test those kind of charophytes, oospores of charophytes which are deposited in this really old sediments, it should be some factors which is responsible for sediment mixing, because without light they are not able to grow. Yes, so
Yes.
so generally only oospores from the really thick sediment layer is
Yes.
able to germinate from the oospores.
Well, Vanja, I'll be very curious to see what the results are from your future analyses of your sediments in that case. It's interesting to think of them as sort of an aquatic complement to what's more often referred to as like terrestrial seed banks. So that was really interesting to hear about. Wonderful. Well, are there any other questions? I think we're good. All set in the chat. Wonderful. All right. So Vanja, if you would stop sharing your screen, and we'll switch
Yes,
over
of
to the
course.
next. Yeah, no worries. We'll switch over to the next presenter. So our second speaker today is Dr. Amit Kumar from the Sathyabama Institute of Science and Technology in India. So as I introduce you, if you'd like to go ahead and start setting up your slides. Dr. Kumar is a marine conservation biologist who received his doctorate from a joint program between the Stazione Zoologica Anton Dorn in Italy and the Open University in the UK. He is currently a scientist at the International Research Center for Climate Change Studies at the Sathyabama Institute of Science and Technology. And today, he will be presenting some of his current research, examining the changes in nutritional content of seaweeds in response to acidification. Welcome, Amit. Thank you so much for joining us.
Hello, everyone. Thank you for the ASLO Amplifying webinar organizing team for providing me the opportunity to share my research work. I think it's showing the network is slightly unstable, so I'll stop my video, if that is okay.
You can go ahead and start whenever you'd like.
Yeah, fine. So, first, okay. So let me give a brief intro where I am located. So I am basically located in I'm basically located in a city called as Chennai in southeast coast of India. And I do study at two different locations. One is Kovalam, which is a sort of sandy beach and with a few intertidal rocks. And the second location is Gulf of Mannar and Park Bay area, which is further down south, very much near to Sri Lanka in the southeast coast of India. And it is a part of UNESCO Man and Biosphere program. So in these two locations, I've been studying what all different organisms are there. So basically marine and the coastal biodiversity and their conservation. Along with that, I am also investigating how the species thriving in these two locations respond to climate change factors, mainly acidification. So as we all are aware that due to anthropogenic influences, atmospheric carbon dioxide is increasing at an unprecedented rate, especially in the last 1 century. Now we have touched almost 424 ppm in last week on 16th April, which is the level of the ppm of the carbon dioxide, which was not experienced by the Earth in at least last 800,000 years. As a result, there is an increase in the global temperature, causing the global warming. And at the same time, the oceanic water is also heating up, and the marine organisms are facing the ocean warming. So ocean act as a sink for these atmospheric carbon dioxide, and at least 1/4 of the atmospheric carbon dioxide enters into the ocean. So what happens is that when carbon dioxide enters into the ocean, it reacts with the water, H2O, and forms the carbonic acid, which further disintegrates into bicarbonate and the carbonate. And of course, there is a release of H plus ion, as a result of which this change in the carbonate chemistry of the seawater causes a phenomenon called as ocean acidification, which you can also understand like another CO2 problem, where the pH and the carbonate ion decreases and the bicarbonate ion increases. In the graph, in the right side, you can see that in the CO2 time series in the North Pacific, as the increase in the carbon dioxide, atmospheric carbon dioxide, it subsequently increases the aqueous carbon dioxide in the ocean, which is correlated with the decrease in the pH of the oceanic seawater. It is having the influences. So basically, ocean acidification is causing more problem to the calcifying organism or the calcifier, as the calcifier undergo decalcification. And at the same time, this decrease in the abundance of carbonate reduces the formation of the cell for them. A decade back in 2013, by Christy Coker et al., has done a marvelous meta-analysis where we can see that most of the studies till then were focused either on the calcifiers and very less studies on the non-calcified, in particular to the macrophytes, including the algae and the seagrasses. And whatever studies were done at that time were mostly focused on the growth and the photosynthesis. Well, it can be understood because this carbonate chemistry changes the or influences the availability of inorganic carbon, which is very much essential for the photosynthesis. However, in the last 1 decade, focus has been given to the other physiological and biological processes as well, because along with the increase in the aqueous carbon dioxide, the ozone acidification also causes the lowering of the pH that can have the negative impact on other biological and the physiological processes. So we have studied in this particular work two species of Gracilaria. We have taken two species of Gracilaria. One is Gracilaria debilis and the Gracilaria folifera. This particular the genus Gracilaria is having more than 100 species worldwide, and many of the species are having economic importance because they are involved in the agar production for the human foods. They are used in the plant nutrients, poultry feeds, and they also have a lot of bioactivity. Because of all these economic importance, many of these species are commercially cultivated and providing the livelihood in the coastal community along the east coast of India. So Gracilaria debilis was collected from a place called as Rameshwaram, very much in the southern part of the country, and Gracilaria folifera from Kovalam. So from two different locations. Upon bringing it to the laboratory, we have acclimatized it for 4 days, and then we set up the experiment independently for both the species in triplicate in the ambient condition. I mean, we expose it to the ambient condition, that is pH 8.1 plus minus 0.05, and the lower pH of pH 7.7. This experiment lasted for 2 weeks in a controlled laboratory condition, and then we analyzed in detail about the different different response variables that includes daily growth rate, primary productivity, pigments, oxidative status, primary metabolites, and bioactivity against the tested pathogen. So basically, what differs in the sea plant than in the land plant is the availability of the inorganic carbon, because in the land plant we have the carbon dioxide which directly intakes by the plants and then for the process of photosynthesis. However, in the sea plants, in the sea you have a lesser concentration of aqueous CO2, which equilibrates with the atmospheric carbon dioxide, or you can say approximately 10 micromole in concentration, whereas the bicarbonate ion is somewhere around 2 millimolar at the surface of the ocean. So if the plant has to do the photosynthesis, it requires this the carbon source. Now, if the carbon dioxide is present in the lesser amount, and if the diffusivity is lower or slower, then it forms as a limiting factor. As a result of which, the sea plants, mostly 95% of all the aquatic plants, have developed a mechanism called as carbon concentrating mechanism, that is the uptake of the bicarbonate ion from the surrounding water, and then that is available for the photosynthesis. If the plants are having carbon concentrating mechanism, it is generally assumed that they may not be carbon limited, and hence the availability, the benefit because of the ozone acidification or elevated carbon dioxide may not be as much as in those plants which are totally dependent on the aqueous carbon dioxide. So first of all, we wanted to understand that both the species of the Gracilaria which we have chosen do have the carbon concentrating mechanism or not. And for that, we have set up the experiment, pH drift experiment, where we have found that the pH raised above 9. And then also we use different inhibitors for the carbonic anhydrase and the bicarbonate ion transporter. And as a result of which we saw that there was a decrease in the rate of photosynthesis, and the pH level raised in those samples where we were using these inhibitors were lesser than the control, thereby suggesting that both the species have the carbon concentrating mechanism. Yet, we found that the primary production increased or primary productivity increased. Most probably, seaweed would use additional aqueous carbon dioxide for photosynthesis and conserve the energy required for the bicarbonate uptake. Now, this increased photophysiology supported the increased daily growth rate because we saw uh, a general increase in the daily growth rate in both the species under the acidified condition. However, the significant increase was observed only for one of the species, that is the dibs. That was for the growth and the photosynthesis. So now that we know that the daily growth rate was increased and the photophysiology or the photosynthesis was increased, we also wanted to see that what is happening to the cells of the seaweeds, whether they undergo any oxidative stress or not. For that, we have analyzed the total antioxidant capacity, lipid peroxidation, and a battery of enzymatic and non-enzymatic antioxidant. And what we have found is that there was no change in the total antioxidant capacity. There was lesser lipid peroxidation under the acidified condition, so it did not increase. And most of the antioxidants, enzymatic and non-enzymatic antioxidants, were comparable for both the species in the acidified and the control conditions, thereby suggesting that both the species do have the or do possess the acclimatization potential under the lower pH condition. Next, we wanted to understand that how the elevated CO2 or decreased pH influences the metabolites. So improved growth was accompanied by the enhanced production of majority of the sugars which we have tested, and the increase in the sugar was could be utilized for various cellular processes such as respiration. And then it also acts as a precursor of the bioactive metabolite. For example, we can see here the increase in the raminos. This could be uh, this could be seen as a enhance in the valuation of both the species, because these raminose sugars have the application or proven application in various industries like pharmaceutical and the cosmetics. If we look into the amino acid, a general increase in more than 28 and 34% was found in Gracilaria folifera and Gracilaria debilis. Uh, and also the essential amino acid was enhanced in the Gracilaria debilis. Now, these increase in amino acid was particularly interested because it could mean that the nutritional advantages to the associated fauna, which were associated with these two species. It also so increase in the sugar and the amino acid could also mean that there could be a stimulus in the bioactivity of the algae. Further, if we look into the fatty acid, there was an increase in the saturated fatty acid around 7% and 10% in Gracilaria folifera and Gracilaria debilis under the acidified condition. This could mean that the algae may need to alter the membrane fluidity for the acclimation under the acidified condition. However, the negative side was the decrease, though not significant, but there was a decrease in the monounsaturated fatty acid and the polyunsaturated fatty acid in both the species under the acidified condition. Now, many of the associated faunas, or we can say the consumers, depends on the algae for their nutrition. This decrease in the monounsaturated fatty acid and polyunsaturated fatty acid can influence the nutritional value, and that can affect the consumer. So this was on the metabolites. Now, when we analyze the algal extract from the acidified condition and the control condition, we found that there was a considerable increase, or we can say that considerable inhibitory activity against various bacterial and the fungal pathogen, which might be driven by the modified primary and the secondary metabolites. We have analyzed in detail the primary and the primary metabolites. However, we have not looked in detail about the different uh, uh, secondary metabolites, in particular to different polyphenols, polyphenolic compounds. which could be attributing to these bioactivity against the bacteria and the fungal pathogen. So in a nutshell, we can see that under the acidified condition, both the species, the growth has increased, the productivity, the pigment composition, most of the sugars, amino acid, and the bioactivity increased. However, the polyunsaturated fatty acid and monounsaturated fatty acid, which could be linked with the nutritional values to the consumers and the associated fauna to the to these two species, are slightly or marginal decreased, which can influence the nutritional value to them. Overall, we can see that from the health perspective, under the acidified condition, both the species have positively responded. And one more thing I just wanted to highlight is that the limitation of the present work, we can see that what we have studied is a single driver experiment and just one pH value of 7.7. We could not do for the multiple pH point, and also the interactive studies with the temperature and the carbon dioxide. Now we are more equipped in terms of the laboratory. So maybe in future we will be continuing the experiment, combining the interactive studies with the different drivers of climate change and understanding the ecophysiological response of the seaweeds. So with this, I would like to conclude my talk, and I would like to thank Mr. Vinu Ganesh, who is a PhD research scholar in my research group, and the funding source, which is SCRB from Department of Science and Technology, Government of India, and the intramural facility or intramural grants for the facilities provided by the institute. So with this, I would like to conclude. Okay, but just before to conclude, I would like to introduce to all the listeners who are in particular from this geographical area, including India, Sri Lanka, Bangladesh, Myanmar, Malaysia, Maldives, Pakistan, and Oman. So we do have a SAROA hub, that is South Asia Regional Hub on Ocean Acidification, for which I am the early career representative. And if you are interested to collaborate or to promote the activities happening in the SAROA hub, please write to us. Please follow us on Twitter, and you can also see that we've been hosting SAROA webinar series. We've been releasing the SAROA hub newsletters. So by any chance, if you are interested to be, actively involved in the participation in the SAROA hub, please do write to us. So with this, I would like to conclude my talk. Thank you.
Thank you, Amit. That was wonderful. Again, if anyone has any questions, feel free to raise your hand or let us know via video, or if you'd like to type it in the chat, please do so. We have time for probably a quick question before our final speaker today. Yes, Danny, if you'd like to go ahead.
Yeah, hi, thank you. Very interesting talk. So I'm a microbiologist, so I might be talking nonsense here. But I have a question. You reported the decrease in unsaturated fatty acids, and at least in bacteria, this is usually correlated with rigidifying the structure of the cell. Is this something that could be related to the same as a response or something in response to the acidification, or what could be the driver of this?
So basically it should have increased, but we have found a decrease in the mono and the polyunsaturated fatty acid. In general, with the other species which we have studied, for example, in Ulva compressa or Sargassum vulgare, it is correlated with the increase in the sugar and the saturated fatty acid, but I don't have really the answer that why it could have decreased in this particular study or for these two species. Yeah.
Thanks.
Wonderful. Thank you so much.